Hello, everyone. Thank you for joining me here virtually today inside my computer screen if you are you, or inside your computer screen if you are me. My name is Rajiv, and today I would like to impress upon you this idea that certain psychedelics could be chemically modifying our proteins in ways that might have long-term impacts on our biology. Upon hearing that, you might have two questions. One, what is a protein? So, proteins are these complex biomolecules inside our cells that are responsible for just about everything, which is why they are sometimes thought of as the machinery of our cells. They can be anything from receptors that allow our cells to sense the world around them, signaling molecules that allow our cells to talk to one another, antibodies that protect us from disease, tiny little skeletal structures that give our cells their shape and form, or even enzymes that are like tiny chemists inside our cells carrying out complex biochemical reactions. Second, you might be wondering, what is a Rajiv? So, Rajiv in its original Sanskrit refers to a blue lotus flower, which I thought some people in this audience might be familiar with due to its serotonergic and psychoactive properties. It is, however, a funny misnomer because there is no such thing as a blue lotus flower. There is instead a blue tropical water lily. Anyways, Rajiv's story starts in about 2019, 2020, when I was watching this really excellent TV show, Hamilton's Pharmacopoeia, and also reading Michael Pollan's popular book, How to Change Your Mind. This, like they did for many people, reignited an interest in the classical psychedelics for me. And around this time, there was also a resurgence of academic literature describing how certain psychedelics, like psilocybin from magic mushrooms, might be useful therapeutics for neuropsychiatric disorders like anxiety and depression. What was remarkable about these studies is that they reaffirmed this idea that a single psychedelic experience could have long-term, long-lasting changes on people's mood and behavior. Since then, there's been a lot of chemical, molecular, cellular, small-scale research on what the mechanism might be where a psychedelic is able to elicit these long-term changes. And that's focused on this idea here, which is that you take a psychedelic, that psychedelic goes to your brain, it acts on your serotonin receptors, and that's responsible for a number of the psychedelic effects you might experience. One of those is neuroplasticity, which is broadly the brain's ability to adapt and change. The overall idea here being that you take a psychedelic, your brain has an opportunity to adapt and change in a way that might elicit a long-term positive benefit on your mood and behavior, your mental state. Now, I decided this was all really interesting. This is what I wanted to focus my own academic research on. The problem for me is that I had just joined a lab that doesn't study psychedelics, never had studied psychedelics, had no intention of studying psychedelics. I had joined a lab that studied eye diseases like glaucoma. Glaucoma is a leading cause of irreversible blindness that's associated with elevated eye pressure. So, high pressure in the eye can damage the eye in a way that first causes loss of peripheral vision, and if severe and left untreated, it can cause total blindness. The lab group I had joined was particularly interested in how parts of the eye become stiff and disorganized during the process of this disease. An example of this disorganization can be seen here. Here we have pictures of the nerve that connects the eye to the brain, fairly important. You can see in a normal eye, you have this crisp, defined structure with lots of space. In the diseased eye, that structure is completely disorganized, completely collapsed, and you can notice that there's parts of the structure that are now sticking together. And that's something we were even more specifically interested in, which is why do parts of the eye during disease become sticky? And it turns out this has a lot to do with enzymes. One of those enzymes is transglutaminase 2. Transglutaminase 2 is probably better known as meat glue. So any chefs in the audience might be familiar with the fact that transglutaminase is the major ingredient in meat glue, which has culinary applications for molding and sticking together meat. What I want everyone to take away from this is one, we live in an exceptionally strange world. And two, transglutaminase 2 is exceptionally good at sticking proteins together. You might recall I mentioned that enzymes are like tiny chemists inside our cells, and this is the chemistry that transglutaminase 2 performs. So proteins are complex molecules that are themselves made up of smaller chemicals. So you can have a protein that's made up of a chemical called lysine. You can also have another protein that contains a chemical called glutamine. What transglutaminase 2 will do is it will take the nitrogen from a lysine, a primary amine, and the nitrogen from a glutamine, a primary amide, and chemically connect them. What you get from this is you have two proteins that are linked together by a stable chemical bond. These proteins are stuck together. You can appreciate that this is important in diseases where parts of the eye are sticking together. Um, Now, I knew that I was going to be hard pressed in a lab that's studying how meat glue makes eyes sticky to pitch a PhD project focused on the mechanism of action of psychedelics. But I thought I might be able to do the next best thing, which is study serotonin. So I think everyone's familiar with the fact that serotonin is the happy neurotransmitter. And importantly, as we discussed, it is the serotonin system in our brain that psychedelics utilize to exert their effects. So I did what any PhD student might do. I Googled serotonin and transglutaminase 2, hoping to find some overlap between my research interests and my lab's research interests. And serendipitously, I came across something called serotonylation. So serotonylation is this transglutaminase 2 mediated process where instead of sticking two proteins together, transglutaminase 2 is now sticking a serotonin molecule onto a protein. This occurs because proteins can have a glutamine with a primary amide, like we discussed, and you can have a molecule of serotonin with a primary amine. And transglutaminase 2 can recognize these two groups and chemically connect them. What you get now is a protein that is chemically modified by a serotonin molecule. You have serotonin stuck onto a protein. Now, this might not seem important, but there's actually been a lot of research suggesting that the serotonylation of proteins can influence their function. This has been remarkably demonstrated with a protein called a histone. So I think everyone's familiar with this idea that DNA makes us who we are. But how your body interacts with your DNA on a day-to-day basis is influenced by its three-dimensional configuration or its three-dimensional shape. And that three-dimensional configuration is dictated by proteins, one of which is a histone. What people have found is that when serotonin molecules are stuck onto histones, they interact with our DNA differently. And what's really remarkable is that people have found that the serotonylation of histones might even be an important part of how antidepressant medications exert their effects. Now, what I want everyone to take away from that is the stable chemical modification of proteins by serotonin molecules could have real and lasting neurobehavioral consequences. And something that's been naggingly on my mind while thinking about all of this is that, well, the classical psychedelics are structurally similar to serotonin. So you can have psychedelic tryptamines like psilocybin from magic mushrooms or dimethyltryptamine DMT that are structurally similar to serotonin. You can also have psychedelic phenethylamines like mescaline from peyote or San Pedro cacti that are structurally similar to dopamine. And I was thinking, well, if serotonin and dopamine and other molecules in our body can stick to proteins, could a psychedelic stick to a protein? Especially a psychedelic like mescaline that has a nice primary amine group. And I started to wonder, is there a way I could study this in the lab? Is there a way in the lab I could research whether or not a psychedelic is able to stick to a protein? And in fact, there is a really interesting way you can study whether or not a protein is being modified by a small molecule like mescaline, but only if you have a certain variant of that molecule. So if we had, say, a molecule that looks just like mescaline, but it has a carbon triple bonded to a carbon at the end of it, an alkyne group or a propargyl group. But my first question was, does a molecule like this even exist? And in fact, it does. I was reading this wonderful book, Phenethylamines I Have Known and Loved, A Chemical Love Story, which is co-authored by the chemist Alexander Shulgin and Anne Shulgin. The first half of this book is a sometimes very graphic depiction of their love for one another. The back half of this book is a description of the synthesis of hundreds of different phenethylamines and their subjective effects. Now, I was flipping through the back half of this book, and sure enough, there is this compound 3,5-dimethoxy-4,2-propanoloxyphenethylamine. And it is exactly what we needed. It is a molecule that looks just like mescaline, but it has a carbon triple bonded to a carbon, a terminal alkyne, a propargyl group on it. The problem for me is that this is not something that's commercially available. I could not buy it to use it in the lab. I don't think many people have even heard of it. But thankfully, I'd always made really, really good grades in chemistry. So me and my buddy Max from the lab got together and we started working on synthesizing this molecule from Alexander Shulgin's book. We spent many long, long days and nights in the lab working on this chemistry. And through that process, we discovered something more important than chemistry, friendship. And through blossoming friendship, we discovered something more important than friendship, chemistry. We took this commercially available molecule, seringaldehyde, we propargylated it with propargyl bromide, which adds our nice little carbon triple carbon bond at the end of it. We then added a nitrogen to it using nitromethane. And then we reduce that nitrogen using lithium aluminum hydride to get a primary amine. And now what we have is this nice molecule that looks just like mescaline, but at the end of it, it has this alkyne or propargyl group. I am going to start referring to this for simplicity as 4-propargyl mescaline because again, it has the structure of mescaline, but it has this alkyne or propargyl group on the 4 position. We know this is the molecule we thought it was because all of these lines here are in the right place. Now, why go through the trouble of making this variant of mescaline? Why did we bother with this? And it's because of this strategy right here. So what we can do with our molecule is we can take human cells that we grow in the lab and we can dose them with our 4-propargyl mescaline. And if the phenomenon we're looking for is really occurring, then transglutaminase 2 that's present in these cells should start sticking our little mescaline variant onto proteins. And what we'll then get is a protein that is chemically modified by mescaline, but with this little terminal alkyne group at the end of it. Then we can do something called click chemistry, which for any chemistry boffins is the copper catalyzed azide alkyne cycloaddition reaction. What this allows us to do is click together 2 specific chemicals. We can take the alkyne group that's sitting on our little mescaline that's sitting on our protein, and we can take an azide like seen down here, which itself is modified chemically by a tracer, and we can click all of this together. And what we then get is a protein that is modified by mescaline that is chemically stably linked to a tracer. In this case, that tracer is biotin. We chose biotin because there is another molecule called streptavidin, which loves to tightly bind to biotin. It loves to stick to biotin. What we can do is we can take magnetic beads that are coated in streptavidin, we can mix them up with our proteins, the streptavidin will find the biotin and stick to it, and then we can use a magnet to specifically pull out proteins from our sample that are modified with our psychedelic, which is stuck to a tag. And then we can examine these proteins further. So next I want to take a second to introduce another character to this story, astrocytes. Astrocytes are these cells that are very important to the function of our brain. They are thought of as the support cells of our brain. They help neurons do what they do. Conveniently for me in an eye lab, it's extremely easy to get astrocytes out of the back of the eye where the eye connects to the brain. So we took human donor eyes and we took astrocytes out of them and we grow those astrocytes on plastic dishes. And then we were able to do our experiments with those astrocytes. The first little experiment we did was just looking to see if we can find our compound going into these cells, if we can find 4-propargyl mescaline in these cells after we treat them. So we dosed our cells with 4-propargyl mescaline, and then we used the process I just described to add a tracer onto our compound. And what we did here is we used a fluorescent molecule attached to a streptavidin, which binds to our biotin. And we're basically able to see red fluorescence. Basically, wherever you see a red spot, that is where our molecule is sitting inside the cell. And what you can see is as we add more 4-propargyl mescaline to our cells, this red gets brighter and brighter. That is a good sign for us that our 4-propargyl mescaline is getting into cells and we can detect it in those cells. Next, I want to take a second to introduce to anyone who doesn't have the misfortune of knowing what a Western blot is, what a Western blot is. Basically, what we can do is we can use our magnets to pull out our proteins that are modified by our psychedelic, and then we can put them onto a gel. We can run electricity through that gel, and that will separate our proteins into these nice defined lines or bands, which we can then use to look at our proteins further. And this is more or less what that looks like. So what we have here is we have taken this gel with our protein separate on it, and we are looking for biotin signal. So that biotin signal looks like black lines on this little gel image here. What we can see is that when we add 4-propargyl mescaline to our cells, we're able to see more and more biotin signal on our proteins. And when we take an inhibitor of the enzyme transglutaminase 2 and add it to the mix, we see that biotin signal start to go away. It gets lighter. This is all a good preliminary indication to us that our psychedelic analog 4-propargyl mescaline is really sticking onto proteins in our cells after we treat them. There's one limitation I want to be clear about here, which is that we are using very high doses of this compound. So whether or not this would really occur in a person with more normal doses of this compound is still an open question for us. Next, what we wanted to do is basically look at these proteins that show this modification and figure out, well, what proteins are actually in this mix. And we get something like this. This is kind of a garbage figure. It's very messy. It's very preliminary. There's a lot of work still required here. But basically what we're starting to see is that there is a broad variety of proteins that might be targets of this sort of psychedelic chemical modification. And what that really means is there's a number of questions we can start to ask about this idea, which is, well, if a psychedelic could really be modifying a variety of different proteins in our cells, could it be having all sorts of different effects in our cells? Could it change the structure of our cells? Could it change how our cells respond to signals? Could it change how our cells manage energy? Could it change how cells manage the space around them? Could it change how proteins are produced in our cells? And really, what I want to have everyone take away from that is this idea that, well, if you feel like your psychedelic trip is sticking around, maybe part of it's in your proteins. Maybe psychedelics are sticking around in your proteins, in your cells, and that is part of why they have such long-lasting effects. There's another takeaway I want everyone to have, which is if you find yourself like me in a situation where you want to research something, but you're not in a lab or a space to do it, just try it anyways, see what you can do. I don't think we've gotten very far with this project yet, but I'm really happy with what we've managed to do, and I'm excited to see where we might go forward with it. If you want the nitty-gritty about this project, we do have a working paper preprint out called Phenethylamine Preliminary In Vitro Evidence for the Covalent Transamidation of Psychedelic Phenethylamines to Glial Proteins using 3,5-dimethoxy-4,2-propanoloxyphenethylamine as a model compound. With that, I would like to thank everyone who has helped me out with this project, especially my PhD mentors, Dr. Abe Clark and Dr. Tara Torra-Vidalis, who have stepped out of their usual research areas to help me with this project, all of my friends and colleagues. I'd like to thank all the PsychedelX coordinators and volunteers for letting me give this talk today, especially Amanda for her helpful advice with this talk, and Dan, the man behind the camera right now. With that, I will say thank you for listening and take any comments, critiques, and questions.